Hi, my name is Ryan Verma. I'm presenting on B-cell targeting treatments for multiple sclerosis. So first, about me. My name is Ryan Verma. I was mentored by Joshua Milstein, and I'm presenting from Houston, even though I'm actually from Phoenix. I chose this topic because of my interest in neuroscience, but also because multiple sclerosis is an important issue that affects millions. First, some intro on multiple sclerosis. So multiple sclerosis, or MS, is a chronic inflammatory demyelinating neurodegenerative disease of the central nervous system in which your immune system attacks the myelin sheath coating your neurons, leading to neurodegeneration, and it's the leading cause of non-traumatic disability in young adults, affecting 2.8 billion people worldwide. Though its causes are unknown, some genetic and environmental risk factors have been identified, such as vitamin D levels, Epstein-Barr virus infection, and tobacco smoking. There are three main types of MS. The first is relapsing remitting, or RRMS, which is characterized by flare-ups in disease activity called relapses, followed by periods of partial or complete remission. Eventually, these relapses will decrease in frequency and gradual progression will take over, and this is called secondary progressive MS. Sometimes this gradual progression occurs from the onset of the disease, and this is referred to as primary progressive MS and is the rarest form, occurring in less than 10% of patients. Next, some intro on B cells. B cells are a type of lymphocyte involved in the immune response. For the development of B cells, they originate in the bone marrow, and then once they're mature, they're released into the bloodstream and gather in lymphoid tissues until they're exposed to an antigen. After exposure to an antigen, they will differentiate into multiple types. First is plasmablasts, which may differentiate further into plasma cells. And both these types of cells produce antibodies, which will attack antigens and lead to them being destroyed. The last type is memory cells, which remember antigens from previous infections, allowing for a more effective and faster reaction upon seeing them again. For B cells' role in MS, firstly, they secrete the autoantibodies that attack the myelin. But previously, it was believed that T cells play the main role in MS, but only recent studies have shown the prevalent impact of B cells, such as their abnormal cytokine production. Cytokines are signaling molecules that can increase or decrease inflammation. And in patients with MS, these levels are abnormal, leading to excessive inflammation. And it is believed that memory B cells play the largest part in this. For my methods, I conducted a literature review using sources found on Google Scholar and PubMed. I used 8 randomized controlled trials that compared certain B-cell targeting treatments to placebos or other treatments, and my original goal was to determine their relative efficacies and safeties to determine which one was the best. For my treatments, first is rituximab, then ocrelizumab, ofatumumab, and Bruton tyrosine kinase inhibitors. First, for rituximab. It is an intravenous mouse-human chimeric anti-CD20 monoclonal antibody, which means it's an antibody produced in mice with many of the regions replaced with human regions to prevent immune responses, and it targets the CD20 protein found on many types of B cells except for plasma cells. For efficacy, rituximab saw a 90% reduction in the number of inflammatory lesions on an MRI in RMS, and that same study also found 50% reduction in the number of patients experiencing relapses. Another study on patients in PPMS found that rituximab led to no difference in the rate of progression. For safety, rituximab led to increased infusion-associated adverse events, or AEs, at the first infusion. However, this decreased with successive infusions. In ocrelizumab, it is an intravenous anti-CD20 monoclonal antibody as well. However, it is humanized, meaning that all regions of the mouse antibody have been replaced except for the one binding to CD20. It is currently approved for the treatment of RMS and PPMS. For its efficacy, one study saw a 45% reduction in the number of relapses per year, and that same study found a 95% reduction in inflammatory lesions. Another study in patients with PPMS found a 17% reduction in the number of patients with confirmed degree disease progression, or CDP, which is a pre-specified increase on the expanded disability status scale that is sustained for 12 weeks. For safety, this also saw increased infusion-associated AEs only at the first infusion, which decreased with successive infusions. Next, ofatumumab. This is a subcutaneous anti-CD20 monoclonal antibody, and it's fully human, meaning that although it was produced in mice, all regions of the antibody came from the human genome. It is currently approved for the treatment of both RRMS and SPMS, and for its efficacy, one study found a greater than 90% reduction in the number of inflammatory lesions, and also decreased the percentage of patients experiencing relapse significantly. Another study in patients with both RMS and SPMS found that it decreased the percentage of patients who had experienced CP. For safety, it had increased injection-related AEs, which also decreased with successive infusions. And lastly, BTK inhibitors. These are inhibitors of the signaling molecule Bruton tyrosine kinase, which is involved in many pathways, including those that control B-cell maturation, activation, and activation of T cells. They're currently used to treat certain B-cell lymphomas, and for their efficacy in MS, evobrutinib was found to have a 70% reduction in the number of inflammatory lesions in RMS, as well as a reduced number of relapses per year. Another BTK inhibitor, tolebrutinib, was found to have a 90% reduction in the number of inflammatory lesions. For safety, the first study on evobrutinib found that it has slightly higher total AEs, mainly due to infections. For my conclusions, first, all 4 treatments are effective at reducing relapses in RMS. BTK inhibitors are slightly inferior for reducing the number of inflammatory lesions in RMS, however, it's still significantly effective. For slowing progression, ocrelizumab is superior to rituximab in PPMS, and ofatumumab is effective in SPMS. However, no data was available on the progressive forms of the disease for BTK inhibitors. Next, the 3 multiple monoclonal antibodies had increased infusion or injection-related AEs that decreased with successive injections, and BTK inhibitors did slightly increase susceptibility to infections. However, none of these are serious safety concerns, and safety should be evaluated on a patient-by-patient basis. Lastly, for my limitations and future research. First limitation was a lack of variety in treatments. B cells in multiple sclerosis is a relatively new topic of study, so there are very few treatments available, and many of them are very similar, such as all the monoclonal antibodies. Next is a lack of available data for the progressive forms of the disease. Often, RRMS is lumped together with SPMS, but they only measure the relapses and not the actual progression. And also, PPMS is very rare, and so it's very rarely studied, so it's very hard to get data on PPMS and SPMS alone. Lastly, differences in data collection made comparison difficult. For example, with relapses, some studies would measure the relapse rate, while some would measure the number of patients experiencing relapses. So though they're measuring the same thing, you can't compare them very well. For potential future research, one would be to test treatments for other types of MS. So these treatments, most of them were only tested for 1 or 2 types, so testing them for all 3 could give us a better idea of their true efficacy. As well, we could test other B-cell targeting treatments on MS. For example, rituximab and BTK inhibitors were originally used for B-cell cancers or other autoimmune diseases. So finding other B-cell targeting treatments and testing them on MS could allow us to find better and more effective treatments for MS. All right, here are my references. And thank you for listening. I hope you enjoyed.